Good morning and happy Mother's Day. I'm Stuart Rosenthal, and it's a pleasure to welcome you to the third in our series of presentations designed to help us better appreciate the presence of Hashem in the world around us and in our daily lives. The Meaningful Tefilah Project at Young Israel Shomrei Emunah offers these free programs to boost our emunah and improve our kavanah in prayer. We welcome KMS as a sponsor of this series. Our first lecture by Dr. Jeremy Schnittman at NASA explored some of the wonders of the universe. Our second one by Dr. Aaron Cypus of NIH addressed some of the amazing organs of the human body. And this morning, we are pleased to present Dr. John Golin of YU speaking about Marabu Ma'asecha at the submicroscopic level, how enzymes work. I will introduce him formally in a moment. Next month, we will welcome Rabbi Yitzchak Breitowitz, Rabbi Emeritus of Woodside Synagogue, who will help us see the hand of Hashem in history. Please mark your calendar for Sunday, June 12th at 10:30 a.m. for his presentation. We plan to continue this lecture series for a number of months to come. If you would like to be informed of upcoming programs and have not already done so, please send your email address to me at stuartrosenthal@yais.org. Please also tell your friends about this series and encourage them to join us or to watch the recordings which are posted on YouTube. I would like to thank the sponsors who support makes these programs possible: Ken and Cheryl Jacobson, Mark and Debbie Katz, Max and Debbie Rudman, and Karen and Daniel Moshinsky. The Moshinskys would like to dedicate their sponsorship of today's presentation, quote, in appreciation of the scientific community for the development of enzyme replacement infusions that saved and still sustain Karen's life, and in appreciation of Dr. Robin Eli, who orchestrated it all and without whom our whole family wouldn't be here today, unquote. Additional sponsors of these ongoing programs are welcome. Please contact me or the Young Israel office. We hope, as COVID recedes into the background in Yirsa Hashem, to be able to return to in-person programming to be followed by refreshments and socializing. We'll keep you informed. This morning's speaker is Dr. John Golin, who fell in love with genetics and biochemistry as an undergraduate. He received a Ph.D. in genetics from the University of Chicago and did postdoctoral work at the Institute of Molecular Biology at the University of Oregon. He says the best thing that happened to him in Oregon was meeting Trish Weissman, to whom he has been married for almost 42 years. John conducted federally funded research and taught courses in genetics and biochemistry for 35 years at the Catholic University of America, where he says both he and the Pope wore kippot. Upon retirement, John and Trish moved to New York City, where he lives across the street from a daughter, son-in-law, and two fantastic grandsons. He says he stays out of trouble by teaching human genetics as an adjunct professor at Stern YU, serving as Gabbai Rishon at the historic 29th Street Shul at Deret El, and staying at least 4 days ahead in Daf Yomi. Now that's impressive. If you have questions for John as he speaks, please type them in the chat window. After he concludes his remarks, my wife Judy will present the questions to John, and he will address as many as time permits. Before he begins, however, I would like now to invite Rabbi Baum Weinberg, Rabbi of Kent Mills Synagogue, to share some words of Torah with us. And let me just spotlight the rabbi. There we go.
Thank you. Thank you, Stuart, for that beautiful introduction. Thank you all for having me. It's an honor to be here with you, and really appreciative of this program and appreciative of allowing KMS to be a co-sponsor and to join you for this program, which has been so enlightening thus far in the first two sessions. And really looking forward, Dr. Golin, to your talk today. Just share a quick thought, perhaps to frame the lecture today. Many people know the Koren Sachs Sidur today has become so popular, sort of a staple in many shuls around the country. But I remember actually before it was published, having the merit to hear Rabbi Sachs, Alav Shalom, come to YU as they were working on this to describe the project of the Siddur. And I remember where he was standing, where I was sitting in the Beis Medrash, and hearing his thoughts about this upcoming publication of a Siddur and a new translation and interpretation. And I was so eager to see what it would be like, which now thankfully we get to hold in our hands. But I remember him making a very salient point, which I wrote down in my notes. I still have in front of me here at the time, when he said a very, very pertinent statement, which I never thought about before, which is that Chazal, our sages, never really wrote a systematic book of Jewish philosophy or theology. You can pick up through the pages of the Talmud, a piece here, a piece there. And medieval Jewish philosophers wrote books of Jewish philosophy, but our sages never wrote a comprehensive book of Jewish philosophy. They wrote halacha, they wrote Jewish law. The Mishnah is really a comprehensive book of Jewish law, but they never wrote a systematic theology. So people say that, Rabbi Sachs noted, but he said they're wrong, because our sages did write a book of philosophy, and that is the Siddur. And in his intro to the actual Siddur, later he published this, and he says, scholars of Judaism, noting that it contains little systematic theology, have sometimes concluded that it's a religion of deeds, not creeds, acts, not beliefs. They were wrong, because they were searching in the wrong place. They were looking for a library of works like Moses Maimonides' Guide for the Perplexed. They should have looked instead at the prayer book. The home of Jewish belief is in the Siddur. It was a phenomenal point. If you look at the structure of the Siddur, you look at the content of prayer, the way the Anshe Knesset HaGedolah and later Jewish sages structured the Siddur, the formalized prayers and liturgy that we have, it speaks volumes to our theology and our philosophy and how we're supposed to think about God and our relationship to Him. In terms of the specific topic today, I think the Siddur very much speaks to the concept of Ma'arabu Ma'aseh Hashem, to the concept of seeing God's hand in nature in many ways. But I want to point to just one. Sukkot Zimra, which we say every day of the week in Shacharis, is really the preparatory stage to the great encounter with God, to the declaration of Shema Yisrael, of accepting God upon ourselves, and the conversation with God that occurs in the Shemona Esrei in the Amidah. Sukkot Zimra is really that stage that teaches us what a relationship with Hashem is about, how to discover that relationship so that we're ready when we reach Kriyat Shema and when we reach the Amidah. One of the central points of Sukkot Zimra is in the Mizmorim that follow Ashrei, the Hallelujahs that follow Ashrei, the Prakim, the chapters of Tehillim Kuf Mem Vav through Kuf Nun that follow Ashrei, really like the heartland of Sukkot Zimra. And if you look in those Mizmarim, you discover something phenomenal. Rav Shimshon David Pincus in his Alav Hashalom Sefer Sha'arim B'Tfilah, is a lesser known work, Sha'arim B'Tfilah. He says the following: Nimsa Sheseder Hazmiros, the order of these Mizmorim, the order of these chapters of Tehillim, Kusulam Mutzav Artzav Aroshu Magi Hashman. It's actually a ladder. It's taking us rung by rung, step by step, training our mind in terms of a philosophy of Hashem in the world. In Birchosav Shalafan of Lachar, the whole structure of Tefillah in general, but specifically these Mizmorim. Od Shar HaPratim, all the details that are conveyed there. Bonim Kulam Yachad et Shlemus HaHekef. These Mizmorim build the overall perspective of God that we are supposed to have. And if you look in these Mizmorim, something really interesting emerges. There's a rung approach, a ladder approach, a staged approach. If you look at the first Mizmor, and you'll take a look later on when you have more time, and I don't have enough time to go into it, but you'll take a look. The first Mizmor after Ashrei talks about Halali Nafshi. My soul should praise God. The next Mizmor speaks about nature. God as HaMechaseh Shamayim Bavim, HaMechim Ala'aretz Matar. God is the one who covers the sky with clouds. He provides the earth with rain. He makes the grass grow on the hills. He gives food to the cattle, to the ravens, etc. Then the next Mizmor speaks about the celestial beings, the Shemesh, Yareach, Koch Ve'or, Shmei HaShamayim. It talks about the nature that's above us. And then in the next Mizmor, it talks about Shir Chadash, it talks about singing about the Messianic era, an era that's not even seen in front of our eyes in the world, a world that doesn't yet even exist. So Rabbi Pincus says, this is actually the evolution of how we're supposed to think of God. We start thinking of God with nafshi, inside myself, the closest circle. Then you go a little wider. You go to the world that's around you, the grass, the hills, the animals, the birds. Then you go a little wider. Wow, you look to the heavens. It's stars, it's celestial beings. And then you go even further and look to a time that doesn't even yet exist. What that structure speaks to for the Hekef Hagadol, what it speaks to about our general approach, our gestalt, our hashkafa about God in the world, is that it's absolutely necessary to look for and find God in nature. The pathway towards that ultimate messianic era starts with the self, but it goes through nature. Nature that's immediately in front of us, nature that's above us, nature that we don't even understand yet. Seeing the hand of God in nature, in the world, in science, in that which we don't even yet know is a critical component of coming to that relationship with HaKadosh Baruch Hu that we all so desire. And this series has really helped us to get there, and I know we'll continue to do so in today's lecture with Dr. Golin, which I'm very eager to hear. Thank you so much for having me, and pass the floor back.
Thank you, Rabbi, for your beautiful and inspiring Divrei Torah. Appreciate that, and we also appreciate your support of this entire program. Okay, John, I'm going to highlight you now, and we look forward to hearing your remarks.
Hi, everybody. It's live from New York. It's me. So I don't know about you, but when I think of Ma'asecha, I think of things like, I don't know, God telling Abraham Avinu to count the stars, or taking a family trip and seeing the Grand Canyon, things that are big and glorious and really show us Hashem's hand. But today I'm going to talk about a world that you don't see, but is working 24/7 and is every bit as miraculous and wonderful. And that's the world of enzymes. So I'm going to share my screen. Let's go up. Yeah. So let's start with some basics. So cells are the basic building blocks of all living things. Cells aggregate into tissues. Tissues aggregate into organs, and organs aggregate into organisms, human beings. There are about 30 trillion cells in an average human being. And as you can see, they come in various kinds. I hope people can see the pointer. Yeah. All cells originate from progenitor stem cells. And everyone is familiar, I'm sure, with what you see on the screen. Do not freak out. Cancer cells are not a normal part of our makeup, but they arise from time to time. And they are usually dispensed with by our immune system. And our immune system originates from white blood cells. In any event, an organism's development and maintenance depends on chemical reactions that take place in these cells. And what do we mean by a chemical reaction? A chemical reaction rearranges the atoms of starting materials, carbon, nitrogen, oxygen, hydrogen, things like that, and converts them into one or more products. And so in the representation, you can think of these colored balls as either being single atoms, carbon and oxygen, or being groups of atoms. And what you can notice is that chemical reactions can do several things. So groups of atoms or single atoms can come together and form larger molecules. And the atoms are attached to one another by chemical bonds. Conversely, and we'll see a bit of this today, you can take larger structures, larger molecules, larger groups of atoms, and they can be broken down into smaller entities. Or, as you can see in the last two examples, atoms can be exchanged between reactants, starting materials, to generate new combinations and new compounds. And in that case, we both are breaking these attachments or chemical bonds and forming new ones. Now, have a look at these numbers. In an average adult, there are 30 trillion cells, just like we said. The number of chemical reactions taking place per second, okay, it's not exact, but it's in the ballpark, in an average adult human being is 37 billion reactions per second. Wow. That's a Jeremy Schnittman number. I mean, that's a huge number. That's a 37 with 21 zeros after it. And if you want to do it on an individual cell level, there are 30 trillion cells. If you divide 37 billion by 30 trillion, you get more than 1 billion chemical reactions taking place in a cell every second. if that isn't Marabu Masecha, honestly, I don't know what it is. It's an incredible number that it's very hard to get your head around that, right? Yeah, okay. So with this background, let's just frame a central problem that we'll be dealing with today. And this is the following. Look, there are thousands and thousands of reactants, right? Starting products in cells. If we want a chemical reaction to take place, the desired reactants have to be brought in proximity to one another. They have to be in the right orientation. And then sometimes old attachments or chemical bonds have to be broken and new attachments have to be made. Now, all this requires a certain amount of input energy. And chemists call this the energy of activation. And they illustrate it with little graphs that you see here. This is a standard in every chemistry high school textbook that exists, I'm sure. So on the left-hand side, on the y-axis, we have a measure of energy. We'll just leave it at that. And then on the x-axis that you see on the bottom, we have the progress of the reaction. You can think of it as time if you want. And so when we start out with reactants, they have a certain inherent energy in them. But then in order to get them to react, you have this huge hill, right? It's called this is the top of the hill and represents from the starting energy of the reactants to the top of the hill is the activation energy. You have to climb this hill in order for the reaction to really get going. And then you produce products that are lower in their energy level, and more stable generally. Okay, now, without any help, it would take a really long time to get the energy from the environment to get to the top of that hill. And biochemical reactions would be much, much, much too slow for life to exist, as we know it at least. And so not surprisingly, these reactions have to be sped up. And as you probably know, that requires something called a catalyst. And what a catalyst does basically is to lower this hill. In cells, the catalysts are enzymes, of course. And this is kind of a deceptive looking graph because it looks like the barrier's been lowered, maybe relatively modestly, but actually I should point out that enzymes can speed up biochemical reactions in cells more than 1 million-fold. They are amazing entities. And so we want to talk a little bit now about what enzymes are. Enzymes are large molecules called proteins. The directions for making proteins come from genes. And as you know, genes are made up of DNA. And DNA is what chromosomes are constructed from. Chromosomes are large DNA molecules that fit into the nuclei of our cells. It's important to understand the relationship between genes and proteins so that we can have a conversation that makes sense to you. And to do this, I'm going to show a short video. So I've showed this video many times. I show it at YU to students. I like it because it's quite succinct and it's clear and it's easy to follow. And just a few points before we look at it. First of all, keep in mind that DNA is made up of building blocks called nucleotides. Nucleotides are put together to make large DNA molecules. There are about 3.2 billion nucleotides in our DNA. A molecule called RNA is also made of nucleotides. The proteins that we're going to be talking about are built of subunits called amino acids. Amino acids and nucleotides are very different chemical entities. So just hold that thought while you watch the film. The film is going to show you AGCT. Those are nucleotides. I don't think the film specifically says that. The second thing I'd like you to keep in mind is pay particular attention to the discussion, especially towards the end of the video, which is only 5 minutes long, about protein shape, because that's going to be incredibly important, and I'll have more to say about that when the video is finished.
Stated Clearly presents, what is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA stated clearly.
Okay, so everyone knows what these are, right? These are Legos, just like the film was talking about. And you know a Lego when you see it. It's not a Tinker toy or a Lincoln Log, which really dates me, I guess. You know they're Legos, right? Because they've got very shiny colors and these little bumps that you see all over. And you can arrange the same set of Legos into different shapes. So the next slide is with sincere apology to my grandsons who are master Lego builders, unlike me, and just to illustrate, right, that these are two different shapes. So obviously the muscle is with amino acids, right? There are 20 different amino acids, and we know they're amino acids and they're not vitamins and they're not sugars because they have characteristic groups of atoms that they share. All 20 amino acids have the same set of similar arrangement of atoms. But they also have atomic differences that give them distinct chemistries. So for example, some amino acids have positive charges on them. Some amino acids have negative charges on them. Some amino acids are uncharged. There are amino acids that like nothing better than sitting in the solution in cells, and there are amino acids that despise it. There are big amino acids and small amino acids, and you get the picture. Now, because these amino acids have different chemistries, they can have favorable and unfavorable interactions with each other. So for example, if you were to try to put two amino acids together that had a positive charge on them, they would repel each other, and that would be a very unstable thing. Similarly, if you tried to put an amino acid that doesn't like being in water or cellular fluid on the outside of the protein, that's unfavorable. In contrast, if you put an amino acid that had a positive charge next to an amino acid in a protein that had a negative charge, that would be a very good thing because that is a state of neutrality, that's stable. And so in general, the shape of a protein, the three-dimensional structure of a protein is the shape that maximizes, through bending and twisting and folding, positive interactions, favorable interactions between amino acids, and it minimizes unfavorable interactions between amino acids. This gives the protein its correct shape, and that's essential to biological function. Now, biochemists have special tools that allow them to look at the shapes of distinct proteins, individual proteins, and they are, as you might expect, extremely varied. So implicit in this discussion is the idea that a protein shape is determined entirely by that particular set of amino acids that's in a protein. And this was demonstrated by a scientist named Christian Anfinsen, who has a particular connection to our community. He won the Nobel Prize for this work. He lived in Kent Mill. And my understanding is he attended the Shomrei Hashkama Minyan many years ago. It's an interesting story. With a name like Christian, you might imagine that he was a convert, and he was, to Orthodox Judaism. And he did his work at the National Institutes of Health. Towards the end of his life, however, he learned that his mother had been Jewish all along. So, the conversion really wasn't necessary in the first place. In any event, what's astounding is how quickly these proteins assume their correct shape. So in the video you saw rather than you heard, that as the protein is being made on the ribosome, it begins to acquire its correct, stable shape. That process takes anywhere from 1/1000th of a second to 1/1,000,000th of a second. It's a truly extraordinary phenomenon. Okay. Having talked a little bit about proteins in general, I want to remind you that enzymes are a set of proteins. They're a major class of proteins, but they're not the only type of protein that exists, right? So proteins do many, many things in cells. For example, hemoglobin is a protein, and it transports oxygen throughout your body. It's not an enzyme. In any event, whether you're dealing with enzymes or whether you're dealing with other kinds of proteins, they have to be in their correct conformation or their correct shape to work. I still would like to get the slideshow to work. I know it's just very frustrating. Anyway, so what I want to do now is turn my attention specifically then to proteins that are enzymes. There are thousands of different enzymes in our cells, and each of them catalyzes a specific chemical reaction, right? A specific set of reactants work together to get a specific set of products. And that means that each enzyme has to be able to look into that pool of all those different chemicals in cells and to pull out just those reactants whose chemistry it's catalyzing. The recognition of an enzyme and the reactants it works with, which we call a special name, we call them substrates, takes place at a particular place on the enzyme called the active site. And the active site is not only where the reactants are recognized, but where the ensuing chemical reaction will take place. So I'm going to show you a picture of an enzyme. This enzyme is called RNase 1. And let me explain a little bit of what it does. So cells are great recyclers. And as you saw in the film, once a messenger RNA is made from a piece of DNA, right, from a gene, that RNA is then sent to the ribosome. And on the ribosome, the directions in the messenger RNA lead to the production of a protein, right? The nucleotides in the RNA are read in sequence, and each of three nucleotides stands for a particular amino acid in the protein, just like the film said. Now, once that process is over, the RNA is degraded. It is broken down into its independent individual subunits or nucleotides, and it's recycled. It's recycled to make new messenger RNAs for other genes and other proteins. That breakdown requires an enzyme, and the enzyme that does it is RNase 1 in the cell. And just by the way, I don't know if you got either the Pfizer or the Moderna messenger RNA vaccine. They injected that into your muscle, and then that messenger RNA was taken up by cells in your immune system. And those immune cells, those are the white blood cells we talked about earlier, they translated that messenger RNA on ribosomes into a protein called spike, which the coronavirus makes. And then those immune cells went to work on that protein to generate the immune response and to give you protection against the virus. That messenger RNA that was injected doesn't hang around. It's immediately destroyed by RNases, RNase 1 being one of them. So these are very, very important enzymes. So let's have a look at this. All these green bumps are the 124 amino acids that make up RNase 1. And only a handful of these amino acids make up the active site. And the active site is seen here holding on to an RNA molecule. So there are a handful of amino acids doing this. And the question then becomes, well, what about all these other amino acids that you see? This looks like a little elephant or a buffalo or something. A lot of these amino acids provide a scaffold or a support, structural support for the cavity, the active site cavity, which is in the interior of the protein. So once the RNA is going to be bound, it's going to be broken down by this enzyme, right? And a lot of people in the past have likened this to a lock and key mechanism. They say, an enzyme's relationship to its substrate is like a lock and a key. You can see I don't like that. In other words, the RNA molecule is going to be broken down as the key. The enzyme is the lock. The key goes into the lock, and presumably the key's broken if we're talking about breaking down RNA. But in any event, this is really not a very good analogy, and I'll explain. I think it's important to understand why. It's all about that hill, that energy hill that we were talking about before. So we said that reactions require a certain input energy, and the whole reason there's an enzyme is to lower that activation energy. And that means that the enzyme has to be supplying the energy that's needed to carry out the chemical reaction. Now, the way that it does this is truly remarkable. It's one of the most amazing things I've studied in science. Instead of having a lock and key, we say that the enzyme and the reactants, the enzyme and the substrate relationship is called an induced fit. And let me explain how this works. Key to understanding the induced fit mechanism is the fact that the enzyme interacts with its substrate specifically. So initially, there are chemical properties of the amino acid in the active site pocket that line up and match chemical features on the particular reactant or substrate. So for example, in the case of RNase 1, its substrate is RNA. There's a positive charge on an amino acid in the active site, that crevice of RNase 1, that can line up easily with a negative charge on the RNA molecule. And so initially, good. The way that then the enzyme recognizes the substrate is through key geometric arrangement between particular amino acids in the active site and particular chemical groups, atoms on the substrate. Now, those interactions are favorable and they release a certain amount of energy that can be used to make that chemical reaction go. So what happens is the following. The enzyme sees its substrate in this pool of chemicals that are in the cell, and it engages it in the active site, but not completely. In other words, there are some favorable interactions that are allowed between the enzyme and the substrate to begin with. that is how the recognition takes place. But now, what happens next is that the enzyme and the substrate engage in a little dance. The enzyme's shape actually moves and changes, as does the substrate. And as this dance takes place, more and more favorable interactions between amino acids in the active site and atoms on the reactant or substrate occur. And as that's occurring, more and more energy is released that can be used to lower this barrier and allow the reaction to proceed, so that you end up at the top of the peak with a perfect fit between the active site, the shape of the active site, and the shape of what started out as the reactant, but is now beginning to proceed towards being the product. So this top of the peak, all the favorable interactions that can take place between the reactant and the enzyme have occurred, and all the energy has been used now to convert what was initially reactant to product. The enzyme then releases this product, and it can do it all over again, which it does, and we'll see it does with a vengeance. So that is how enzymes really work. It's a miraculous, wonderful thing called the induced fit. Okay, I hope you have some idea then of the beauty of an enzyme interacting with its substrate and the wonders of proteins. And I'd like to talk about some of my favorite enzymes. And the first one I'm going to talk about is one called chymosin. Everybody in the room says, I've never heard of chymosin, but you have heard of chymosin because you've heard of rennet, right? Rennet is used to make hard custard and to make hard cheese. And the key component of rennet is an enzyme called chymosin. The chymosin is normally used to digest milk. So chymosin's reactant is called casein. Its substrate is casein. It's a protein that's present in milk. And when you digest milk, you begin by breaking down casein. And what chymosin does is to take this milk protein and literally cut it in half. The protein is 212 amino acids long, and chymosin snips it at amino acid number 105. And that creates the sort of clumping that's on the way to making a hard cheese. Now, rennet is found in the stomachs of animals that use milk. So, for example, like this cow. This is not an ordinary cow. This is a near miss on a red heifer. Are you able to see, are you seeing the cow? People seeing the cow?
Yes.
Good, okay. So this is an almost red heifer. The problem was that it made a patch of white hairs up here. I see it where the pointer is. A little patch of white hairs made the heifer puzzle. However, I'm sure its chymosin gene and its chymosin protein work just fine. And I was feeling kind of biblical. So I just want to remind you that camels have chymosin gene. They make milk too. But I'm not sure I'd want to get anywhere near that animal to try to get at its rennet. Now, as everyone knows, reams and reams have been written on the kashrut of rennet, right? And in Masechet Avodah Zarah, Daf Lamed Hey 35, bottom of Amud Aleph, there's a whole sugya on Gavinus Akum, Gentile cheese. Hint, it's not kosher. So the issue is a machloket between Rav Huna and Shmuel. So Rav Huna says, the reason that you can't use Gentile cheese, Gavinus Akum, is because sometimes they use animals that aren't kosher, and when you get the rennet out of these animals, there are particles in milk that are in there, and when you mix that with your cheese, of course, you have unkosher cheese. And Shmuel says, Well, the problem is, look, you could use a kosher animal, but it's an availance. It's not, it's shechted by a Gentile, and that automatically makes it no good. So that's the reason that Gemilas Akum is not kosher. Gemara then goes on, it's really interesting, and says, you know what? You can't even eat Gentile cheese made from tree, in which the rennet comes from tree sap. Okay, so the last time that I checked, trees don't make milk. They don't even, and they don't make money either for that matter, right? So what's going on here? And the answer is that although trees don't make milk, trees have proteins that have chemical properties, much like casein, the one that rennet really works on in milk. And they have enzymes that function similarly to chymosin. So you can make cheese using the tree enzyme. The Gemara says the problem with that is that you have an issue of orla, which is kind of interesting because orlas usually refers to fruit. What about tree sap? The Gemara says, tree sap is like fruit. So interestingly enough, Jews were among the first to develop something called thistle cheese, because trees aren't the only plants that have enzymes resembling chymosin. All plants have enzymes resembling chymosin. And in fact, microbes, bacteria, have enzymes that resemble chymosin. And most of the kosher cheese being made now is actually made with microbial, it's called microbial rennet, an enzyme then that can work on even casein in milk. So I think it's interesting to see that. Now, what about our chymosin gene? It's on chromosome 1, but it doesn't work anymore. It's been supplanted by other enzymes that are in our digestive system. And there are a lot of them. My goodness. The next time you eat a calorie-laden Shabbos meal and your enzymes are going to be working 24/7 to break down that food. And as you can see, right, there are a lot of different kinds. There are enzymes that break down starches. There are enzymes that break down carbohydrates, so on and so forth. Chymosin is a type of enzyme called a protease. It breaks down protein. And we have a lot of different proteases that do that job very, very well. We're Jews and we like food, but digestive enzymes are just a drop in the bucket. There have been about 3,500 human enzymes fully studied, and there are plenty more to go. So if you're interested in biochemistry, there's plenty of work left to do in understanding the various enzymes and how they function in our body. One of the things that I think is important to realize is it takes enzymes to make enzymes. So every single time that a cell divides, it has to copy its genetic material from scratch to generate a whole new set of genes for the new cell that's being made. And that means that when the DNA is being copied, it's got to hook those nucleotides together, right, to make one long DNA molecule and to make those DNA molecules into chromosomes. That doesn't get done by itself. It requires enzymes to do that. And the major enzyme that does that is an enzyme called DNA polymerase. To just give you an idea of what's going on here, it takes about 6 to 8 hours to copy all of our DNA, to hook those, to make those chemical bonds between nucleotides, which means that we are making about 7 million of those connections per minute, which is an enormous number. And that's not all. If you take the genes and you want to make proteins from them, you first have to make that messenger RNA that the film talked about. That requires enzymes. You're putting nucleotides together again. Those are the enzyme involved there is something called RNA polymerase. And finally, when you're turning that RNA, when you're decoding it and reading it on the ribosome, That requires an enzyme. To hook amino acids together requires an enzyme called transpeptidase. So what came first, the chicken or the egg? Did the enzyme come first or did the gene come first? It's beyond my pay grade, folks, but it just goes to show you, right? There are enzymes doing everything, including making genes. Finally, I'd like to close with one of my favorite enzymes. Way back, it's actually 2 years, it seems like eternity. When we began with the pandemic that we're still in, everybody went racing to the store and there was nothing on the shelves, but everybody wanted bleach and hydrogen peroxide because in the early days of the pandemic, it was felt that you needed to disinfect all the surfaces in your house and UV treat your food and things like that. And so my local Target quickly ran out of this hydrogen peroxide, which they usually stock. So hydrogen peroxide's nasty stuff. And of course, it'll kill coronavirus. And if you drink it, it'll kill you too. We make hydrogen peroxide. This is remarkable. Just as part of our basic cellular biochemistry, we make small amounts of hydrogen peroxide. And if that hydrogen peroxide were allowed to build up, we'd have organ damage and eventually death. So surprise, surprise, there's an enzyme to take care of the hydrogen peroxide that we build up in our body. And it's given a name called catalase, which I think is a boring name. It just means catalyze. So that all enzymes may do that. So what does catalase do? So catalase takes the hydrogen peroxide that's building up in our cells and it converts it to water and oxygen. Perfectly harmless and in fact usable entities that the cell can certainly use. Here's the remarkable thing. A single copy of catalase, and there are multiple copies in your cells, converts, look at this number, 2.8 million molecules of hydrogen peroxide per second. That's a number that I find, again, marabu masecha. I mean, it's a number that I just can't even begin to conceive of. I mean, if you buy a fancy washing machine and you set it on the high-speed spin, it goes 20 revolutions per second. This is 2.8 million. So I think I'll land my plane here, and I'm happy to answer questions that you might have.
I'm sorry.
Thank you so much, John. Oh my gosh. I think everybody is so overwhelmed with the facts that we don't have a lot of questions in the chat, but I have a couple of questions for you. And thank you so much for sharing all of this with us. I guess one thing that keeps coming into my mind and Stuart's mind is, and I don't know if there's any way you can even begin to address this or answer it in a brief way, but obviously there's so much we still don't know, but there's an awful lot that we do know. And for example, like the example you gave, the vaccine wouldn't work if we didn't understand so much about how all these processes happen. But I mean, how do scientists even begin to approach these things that are not visible and it's such a remove from us?
So the whole study of enzymes, begins, you don't look just at a single picture of a molecule, and the properties of enzymes are deduced from getting them in pure form in a test tube and studying the chemical reaction in detail. You're asking a very complicated question that is kind of hard. You infer a lot about an enzyme, not from the way it looks, but from the way it acts, in an assay. for that enzyme. You can measure, how quickly it makes a product, for example. But the whole history of the vaccines, I think this is really important. This is a subject that's so politically tinged. It's a shame. So much of the work that went into these vaccines is the result of years of analysis and study. So people, one of the things that was hard was, they're developed too quickly. But in reality, they weren't developed too quickly. They relied on really groundbreaking work that had been going on for many, many years. I'm not sure that answers the question, but.
Thank you very much. Okay, thank you. Jeremy Schnittman wants to ask a question too. Jeremy, you want to unmute yourself?
Yep. Hi, John. Thank you so much. It's a great talk. So you mentioned how catalysts can speed up reactions and they lower the activation energy. Are those two like exactly synonymous, or can you have things that speed it up but don't lower the energy, or lower the energy and don't speed it up but kind of slow it down? Or
no, lowering the activation energy means speeding up. There's not, there might be other biochemists listening who beg to differ, but the major problem is that need for energy to get a reaction going, and the only way around that is to lower that barrier in order to get the reaction to go faster.
Okay, and maybe a corollary is, are there any reactions that we actually want to slow down, and there are enzymes that do that or prevent certain reactions from taking place?
Oh, sure. There are enzymes that can let me just think about a question. See if I can come up with an example.
John, center yourself more towards the microphone, please.
Okay. So there are ways of slowing down an enzymatic reaction through inhibition. So what often happens is you'll have a series of enzyme-driven reactions to generate products. And when sufficient product has been made, that product acts as what's called an inhibitor of the enzyme. And so the process then slows down and even stops. There are also enzymes that stop processes by creating products that are endpoints in reactions. But usually you're talking about generating products. Now, so for example, the fact that the RNA molecule that's chewed up by RNase 1 doesn't hang around longer is a way of controlling how much protein from that RNA is being made. So the answer is, I guess, yes.
Okay, thank you.
Make any sense?
Yeah, great. Thank you, John.
Thank you. Are there other questions?
Okay, well, thank you all for joining us. I apologize for my disappearance earlier. I'm glad we got the recording restarted. This will be posted on YouTube as soon as possible, sometime this week. Thank you, John, very, very much. Thank you, Rabbi. Thank you all. And we'll see you hopefully on June the 12th.